protein that allow for the steroid signal. So this is an inducer signal to make the regulatory protein change its conformation so that it can then, using this nuclear localization signal, get in, bind to the DNA, and alter transcription levels. Okay? Good? Are you okay? All right. Now, the next, we still haven't left this particular spot yet, so we're still DNA to RNA. And so we talked about a couple other examples of strategies for still deciding whether or not the DNA is going to get transcribed into RNA. Or I should say into messenger RNA is what we're deciding here as well. And so since we're now talking about eukaryotes, one of those examples is, yes?
For the protein hormones, do you just want us to know the exact same thing except that the receptor is attached to the outside of the cell? And
Yes,
get in?
that's right. So for protein hormones, the protein hormones don't actually get into the cell. They have a receptor that's at the surface of the cell, and the interaction of the hormone with the receptor at the surface of the cell initiates a signaling cascade that changes transcription. That's all you need to know about them.
How does the transcription activation domain come into play there? In the protein?
For the protein hormones? So there what happens is, because of the signaling pathway, eventually a transcription factor gets modified in such a way that it can then change gene expression in the nucleus. And so it's got a transcriptional activation domain, that transcription factor, but until the signaling cascade is initiated and completed, it's not able to change transcription levels. Okay? And then you had a question as well?
Oh, yeah.
You'll be next, but you're certain.
Wait, so where is the steroid hormone receptors? Where?
In the cytoplasm.
Oh, okay.
So it's not in the nucleus, it's not at the surface of the cell, it's in the cytoplasm.
Okay.
So it's sitting around waiting for the steroid to get into the cell. And the steroid can do that because it's soluble in the lipid membrane. Okay? And you had a question?
Should we know about glucose pathway and tyrosine kinase? Okay.
I mean, you can if you want. But yeah, I mean, it's good information, but the specifics I wouldn't worry about. I mainly wanted that as an example of a protein hormone and how that interacts with the receptor, and then what happens after that. Okay. All right. So let's see. All right. So in terms of another example, so this is going to be actually example 3. We can have what's called alternative splicing in eukaryotes. And this is still at the DNA to messenger RNA stage. So the example we talked about was amylase, mouse amylase. And although you did not get to do this and won't get to do this in the lab, usually we have a lab where you spit in a test tube. We're not doing that lab for the rest of the semester. But you'll get some kind of a, do it online kind of lab. That's very computer. Yeah. You can spit on your own computer, just don't share it with anybody else. All right, so mouse amylase, you got the salivary form and the liver form. And the way that you get them to be different is by how you put together the exons. So the salivary gland version might have exon 1 plus exon 3, whereas the liver form has exon 2 plus exon 3. And the result is when you get these messenger RNAs, you get a different form of the protein. Okay? So that's the example that we talked about for another way to regulate transcription by alternative splicing. Okay? And let's see what else we do. Now that's actually that is after you, I'm sorry, I just
skipped
led you
over
guys
the
astray.
DNA and
Yeah,
methylation.
no, I just led you guys astray, sorry about that. It's all being recorded for posterity. This actually is over here, so you get your initial, that's right, this is later. So this is in the RNA to messenger RNA stage. Sorry about that. In this stage here, this is going to be later. So actually 3 is DNA methylation. And that's still at that stage. So DNA methylation can affect, it's going to decrease transcription levels. And we talk about 4 different examples where that can take place. And so that is in the case of fetal versus adult genes, cell differentiation, X inactivation, and also genetic imprinting. In each case, the pattern of methylation of the DNA determines which genes get expressed and which genes don't get expressed. And methylation in general tends to reduce levels of transcription. So when an individual switches from being a fetus to, I don't know, live birth, if you want to call it that, the fetal genes are turned off by methylation. The adult genes are not methylated. When genes that determine whether or not precursor cells are going to be muscle cells or kidney cells or whatever, when we turn off the other genes and only allow the muscle cells to differentiate, those other genes that are turned off by methylation. During X inactivation, which we'll talk about more after the exam, genes on one of the X chromosomes are turned off if you have more than one X chromosome. If you don't have more than one X chromosome, then you don't do X inactivation. And then finally, genetic imprinting has to do with the maternal and paternal patterns of methylation that they provide to the gametes to control how the individual develops after fertilization. Okay? And all of those have to do with methylation. Okay? Any questions? Yes?
Are you gonna go into sickle cells? I don't ask it if you aren't.
Well, so sickle cell is an example where the adult form of the gene is not functional properly, whereas the fetal form is. And so that was just an example where we know that if we were to activate the fetal gene, it would work. And the way we could activate it is by relieving the methylation of that gene.
Okay. I was confused about the overall topic of DNA methylation because I thought it was when a methyl group is attached to a cytosine.
It is.
But then when we talk about 5-AZO-C, we talk about a methyl group not being able to attach to the cytosine.
That's because 5-AZO-C is missing the substituent or the group where methylation could occur.
Yes. But then that decreases transcription then on the adult gene.
That would decrease, that would decrease methylation.
Methylation?
Yeah, so if you take that analog, 5-azocytosine, and you put that into DNA, then every place that goes in is a place that can't be methylated. And if it can't be methylated, then a gene like the fetal form of hemoglobin could now be expressed because it's not methylated anymore.
Okay.
That was the point of that.
Okay.
Was just to show that if we artificially disturb the methylation patterns, you'll get expression of genes that aren't supposed to be normally expressed.
Okay, and then with sickle cell anemia, when you're changing from an adenine to a thymine, in the point mutation, is that what changes for sickle cell? When you have
You're changing, I don't recall which nucleotide change it is, but it's a single amino acid change that results.
How does that, since it's not changing does it change then the methylation rate of the cytosine's capability?
No, not at all.
Then how does that then alter the ability for the adult gene to be expressed in the fetal as a relation to DNA methylation?
Okay, so we're mixing together a couple of things. So in an individual that has sickle cell anemia, they have a defective adult form of hemoglobin gene. And that hemoglobin it causes a misshape in the red blood cells. That adult form will be expressed as soon as the individual is born. The fetal form, while the individual was in utero and was just a fetus, not just, when it was a fetus, the fetal form was being expressed and the adult form was not yet being expressed. Again, probably because of methylation patterns, but actually I don't know in that instance if the adult form was methylated in the fetus and then unmethylated in the adult. I'm not sure about that. What I do know is that when the fetus is no longer a fetus and it's become the baby or whatever, in that situation, the fetal gene is methylated. So it's not expressed. And the adult form is expressed, and that's what the problem is. So the problem with sickle cell is that there's a change in the hemoglobin, not that the adult form is, well, it's a problem that the adult form is being expressed because it doesn't work. But in those individuals, if the fetal form were to continue to work, then they would be okay. In fact, there's some treatments that are being proposed to try and reactivate the fetal form of hemoglobin in those individuals. Not in anybody else, but just in those individuals. Because they could use that form, they could use their own gene that way, and then they would not have the same problems.
Okay.
Does that make sense?
Yeah.
Okay, yes.
I have a question. So back to the sickle cell anemia. So anyone who does have sickle cell anemia, is their adult hemoglobin genes methylated?
No, it's not that they're adult, it's not that the adult form is methylated, it's that the adult form is defective.
Oh, it's defective.
Yeah,
So
it
methylation-
encodes, methylation of that is not the issue.
Okay.
It's that it encodes a gene with a missense mutation. It's got a point mutation leading to a wrong amino acid.
So methylation really doesn't have to do anything with sickle cell anemia?
No, it doesn't. It doesn't have anything to do with this sickle cell anemia other than the fact that the fetal form, which is a functional gene, is turned off because of methylation.
So the fetal form is going to be methylated
Yes,
when the fetus is born?
yes.
Okay.
That's true in people that don't have sickle cell anemia as well.
Yeah, that's what I was
Right, our
confused.
fetal form is also turned off. Those of us that don't have sickle cell anemia. Our fetal form is turned off by methylation. Our adult form is not methylated, and we express it, but our adult form works, and the sickle cell form doesn't work.
So therefore, whether you're an adult or whether you have sickle cell anemia or you don't, your fetal hemoglobin gene will be methylated.
Yes.
Okay.
Yes, question.
So by bringing this up, like the fetal form methylation versus the adult form, you were proposing a possible solution to sickle cell anemia. If we could demethylate that fetal form, then we could solve the fact that we have a defective or adult form, because now we have a.
Yeah, although I think
Eliminate DNA methylation.
I think the more general reason I brought that up is
Okay, that's interesting.
that if you were to perturb methylation, so if you were to use 5-azocytosine to change the methylation patterns, you would see a lot of things happen. In an individual that has sickle cell, you'd see, or us, you would see the fetal form of the gene expressed.
Mhm.
Now, for a sickle cell individual, that would be good, right? Because that would work. For us, I don't know that it would matter.
Right.
But the problem is, that's not the only thing that happens when you do that, right?
Right.
So you get the fetal form expressed. You also start changing cells that are precursors for something else that now start becoming muscle cells because you've taken away the control of that particular thing, you start seeing expression of genes on the X chromosome in copy numbers that you're not supposed to express, and you also perturb genetic imprinting. So I would not propose that that would be a good way to treat sickle cell anemia. But what they are doing is considering ways just to turn on the fetal form of the gene in individuals that have sickle cell. And it doesn't have to do with methylation. They've got other ways to do it.
Okay.
Yeah, so it was mainly that, this just shows you how important methylation is in terms of controlling gene expression. That was the main take home.
I got it.
Yeah.
Okay? And I hope everybody, all of the participants at home, I hope you got that too. All right, so we talked about that, and then still in the correct zone, which I wasn't a minute ago, the other thing we talked about was reversible DNA amplification. Reversible and selective DNA amplification. So this example was a case of in xenophus or frog oocytes. And it has to do with the fact that at a certain stage of development, they need lots of ribosome. And so they will produce lots of ribosomal RNA. And the way that they do that is they take the region of DNA where there are ribosomal RNA genes, and they make lots of copies just of that region of DNA. So they increase the number of copies of the ribosomal RNA gene. And it's just ribosomal RNA genes. And during that period of development, that way they get more expression of those genes, so they can get lots of ribosomal RNA. And then after that period of development, the number of copies shrinks back down. Okay? That makes sense? All right. So all of that, except for this mistake I made about when alternative splicing occurs, all that takes place from the DNA to RNA decision-making point. The alternative splicing is now in section B, where we have the RNA, and the question is, do we make a particular protein? So alternative splicing affects what types of RNAs we get, which affects what types of proteins we get. So at this stage, we're looking at this whole transition controlling the translation products, or the making of translation to give us protein. So alternative splicing is controlling whether or not a particular type of messenger RNA is produced. And the example was the salivary form of amylase versus the liver form. We also discussed, and so this is going to be in eukaryotes. We also discussed antisense RNAs. also over here we'll have and the idea was that, and this is something that could be in prokaryotes or eukaryotes. And in this situation, you have your messenger RNA here, and you have another RNA which is antisense to it. And the interaction of those two blocks translation of a particular target messenger RNA. Okay? And we didn't talk about any specific examples of that, but that is another strategy for controlling expression. And it occurs in this stage between RNA and protein. Okay? Questions about any of that? Yes?
Is translation blocked because the Shine-Delgado sequence is blocked here? Or
That's a good question. So the question is, why would translation be blocked? So one way to think about it is that the Shine-Delgado sequence and or start codon are blocked. So the ribosome can't use that as a way to start translation. It also turns out that oftentimes in cells, double-stranded RNA gets cut. And so there are also enzymes in the cell that when they see this will cut the RNA. And that also interferes with translation. Okay. And then the last example that we talked about in this area was the example of R17 phage. If you recall, R17 phage has a single-stranded RNA as its genetic material. And on it are 3 genes, A, code protein, and replicase, of which you have to have 1 code protein molecule for every phage, but you only need 1 replicase molecule for every 100 phage. I just made up that number. But in other words, you need a lot more code protein than you need replicase. So replicase is an enzyme that makes RNA copies. So it copies the genome or the genetic material. Code protein covers RNA copies. It becomes the protein code for the phage. So you need a lot more of this. So once you have enough replicase, you also have enough code protein that some of it can cover this part, which is the Shine-Delgado sequence for replicase. And if it covers it, so if the code protein covers that, that's going to block translation of the replicase gene. By covering that region where the ribosome would normally bind. Okay? Questions about any of that? Okay? Unfortunately, I can't ask the audience at home, but the next and last area that we talked about was the strategy where you've got a protein and you control the phenotype. And one example we looked at was called end product or feedback inhibition. So in this case here, you can control what happens by end product or feedback inhibition. And that was the example where we had an enzymatic pathway, and the last compound that's produced in the pathway could interact with one of the previous enzymes in the pathway and physically prevent it from synthesizing more of itself. Does anybody need to go over any of that? Questions about feedback inhibition or end product inhibition? Question?
I have a question. So the R17 phage, that's just prokaryotes.
Yes, our 17 page is only in prokaryotes.
The antisense one, that's prokaryotes or eukaryotes?
Correct.
And then
Alternative
lysine
splicing,
is just eukaryotes.
just eukaryotes.
And then the end product feedback, that's both as well.
That's both as well.
You said antisense control is both probes and.
It can be found in both. The cases I'm most familiar with are in prokaryotes, but I think there are examples, whose specifics I can't tell you, but also in eukaryotes.
Do we need to know that for the exam? Like, which
Yeah.
of these pertain to?
Not as much. I mean, in that case, since it can work for both, I wouldn't worry about it too much. But our 17 phage is clearly prokaryotes, and alternative splicing is clearly eukaryotes. Okay. All right, that's all the regulation. See, that didn't take too long. All right, what do we got left? Well, what we got left are mutations, right? So now we're on mutations. So I'm going to erase all of this. And we sort of, at the very beginning, talked about different kinds of mutations, so that gives us a little bit of a head start. So this would be mutations. Okay, so remember mutations, this has to do with changes in the primary genetic material, either for a cell or for a viral type entity, okay? So in cellular organisms, that means it's going to be what? What? DNA, that's right. So this is going to be DNA for cells, DNA or RNA, depending upon the type of virus. So coronavirus, for example, mutations in that would take place in, does anybody know what kind of virus coronavirus is? RNA virus. So coronavirus, the mutations would be in its RNA. Herpes virus, that would be in its DNA, because herpes virus is a DNA virus. Okay? Just like phage T4 is a DNA virus. Okay. All right. So where are we at? Yeah. So that's mutations, and then a mutant is going to be the product of a mutation. All right, and we said that there's a typical sort of spontaneous rate of mutations that has to do with usually the error rate of DNA polymerase and how well that's fixed. Yes?
So does the coronavirus have like a reverse transcriptase?
No. So retroviruses have reverse transcriptase. So HIV and viruses that are related to that, they have a way to take their RNA, copy it into DNA, and then put that into our genomes. Coronaviruses, flu viruses, I'm not sure, there's some other RNA viruses that I'm not familiar with, but they don't necessarily get their RNA converted. Instead, their RNA gets immediately translated, or it gets copied into a form of RNA that then gets translated. So they don't have a DNA intermediate, some of those viruses. Yeah, cool stuff. Okay, all right, so that's mutations, and then what we spent one part of the time talking about were Point mutations. And, I will emphasize this over and over again, these can revert. And they revert at approximately the same frequency at which they appeared. They also map to a single genetic location. And of course, they involve one nucleotide or one base pair change. Okay? So that's how you know you're talking about a point mutation. Mutations that involve more than one nucleotide change are not point mutations. They pretty much don't revert, and they don't map necessarily to a single point on a genetic map. Okay? Now, we said that there are both base substitutions that's just exchanging one nucleotide for another versus frame shifts. And that's either an insertion or deletion of one base pair. Okay. The other thing that we talked about was that there are, as a result of these kinds of point mutations, there are different kinds of specific changes that occur typically when we're talking about proteins. Okay, so we have a nonsense mutation, missense mutation, and silent mutations. And then we can talk about their effects on phenotype. So nonsense mutation is where a codon for an amino acid is changed to a codon for stop, or a stop codon. Hence the name nonsense, because the codon you get is a nonsense codon. Whereas missense mutation, you have a codon for an amino acid is changed to a codon for a different amino acid. And then silent mutation is a change in genotype without a change in phenotype. Okay. So in terms of the effect on phenotype, no change is observed. For nonsense mutation, what do you think is likely to happen as far as phenotype?
Short protection.
Yeah,
Non-functional.
yeah, it's likely to be non-functional. Particularly if we're talking about a protein. Now you can get a nonsense mutation in a molecule that's not normally translated, and that won't necessarily affect its function at all, right? So